Well, let's go ahead and get started. Hi, everyone. Thank you for joining us for the 2024 First Flush Volunteer Training. We're super excited to have you all here with us today. And on behalf of everyone in the RCD team, I just want to thank all of you for your time and commitment to this important program. This is something that we really can't do without all of your support. Um, and it's also something that, we hope you can all learn something about water quality through your participation and, just another opportunity to build some communities. We really appreciate it. And before we get started, I want to start with just a few housekeeping notes. First of all, as we just said, this presentation is being recorded for the folks that aren't able to join us today so they can watch it later. And second, we welcome and invite questions, and we'll have time for questions at the end. So we ask that you please kind of save your questions till the end. But if you're worried that you might forget them along the way, you're welcome to just drop them in the chat as well, which there should be a chat button on the bottom that you can click, and that'll open a little panel on the right-hand side. So you can drop your questions in there, and we'll get to all of them at the end. So to kick off today's training, I think it would be great if we started off by getting to know each other a little bit. And since we have a good number of people here, I think we'll do that in the chat as well, and that'll be a good practice. So if you could please put your name in the chat and then describe either what inspired you to volunteer with this program and or something you're hoping to learn through your participation with us. So we'll give just a minute for that. And while we're doing that, I think the Resource Conservation District team will do our introductions. So hi, everyone. My name is Clifton Herman. I'm a water quality specialist with the San Mateo Resource Conservation District. I started this position in June, so I'm a little bit fresh. I'm really excited to be here. My education is in marine biology. I have experience working in private consulting, as well as state and local governments on water quality and conservation related projects. I also spent some time as an outdoor educator. I'd say the thing that drew me to this type of work is that I have a strong passion for the natural environment, and I always strive to build community through the work that I do. So I'm really excited to meet all of you. I'm really excited to be a part of this program.
Hello, everyone. I'm Grace Allen, and I'm a water quality project manager at the RCD. I've been here for about a year and a half now, and I did this program last year, so I see some familiar faces, and I'm excited to get to meet everyone. So thank you so much for being here.
Uh, awesome. Yeah, thank you for all these great answers. Yeah, monitoring for water quality pollutants is super important for people, plants, and animals. Totally agree. Cool. So let's hop right in. So for this training, I'll just start with a little overview. So first we'll cover what the First Flush program is, and we'll talk a little bit about like what a watershed is. Then we'll describe what we test for in this program. And then we'll kind of get over into what volunteering entails with us. We do this project by breaking all the volunteers into teams. So we'll talk a little bit about what that looks like, and what information we need from you to kind of set that all up and how to get things going. We'll talk about what you can expect on the day of the actual sampling event. And then we'll cover a little bit about sampling materials and protocols and things like that. This is a two-part training. So this first one will kind of introduce you to all of these topics. And then the second part of the training is in person, where we actually, collect samples. So that's, if there's anything that you're not totally clear on today, that's totally okay, because we'll cover that in the next one. That next training will also have a video available if you're not able to make it in person. Um, and then, of course, we'll talk about some safety tips for, collecting your samples safely, and we'll have a summary at the end of all of it. So, what is, we say RCD a lot, we throw that around. It stands for Resource Conservation District. So, what are we? What are we doing? Some of you may already be familiar with the RCD since you heard about this program, but if not, we are a special district, or in other words, a form of local government formed by the community to provide them with services. And in our case, we were formed in the 1930s to help people help the land. There are about 100 RCDs across the state serving as local hubs for community-led conservation. And in San Mateo County, we focus on these focal areas. So that's water, climate, wildlife, agriculture, and fire and forest health. The First Flush program is managed by our water quality team, which is Grace and myself. So you're looking at us. And but as you can expect, these program areas are in many ways interrelated. And we often call on the expertise of our colleagues to find creative and collaborative solutions to help people in our community. And so what is First Flush? Despite what this name might suggest, the First Flush program kind of thankfully has very little to do with toilets or sewage or anything like that. Instead, this program focuses on measuring water quality in the region during the first big rain of the year when pollutants such as oils and brake dust, tire dust, things like that, that have accumulated on roadways and sidewalks and other impervious surfaces throughout the dry season get washed into waterways, creeks, streams, and eventually the ocean. So this is a very long-running program going all the way back to the year 2000 when it was first started. What this means is that the First Flush program has created a very valuable long-term data set that we can use to inform actions that protect and improve water quality throughout the region. The RCD manages this program for San Mateo County in coordination with the Monterey Bay National Marine Sanctuary, because all the coastal creeks in San Mateo County flow into the Monterey Bay National Marine Sanctuary. Something that's super exciting about this program is that Monterey County also has their own First Flush program that the sanctuary manages, which means that we can actually use the data that we create through this program to learn more about our local conditions, but we can also make regional comparisons across multiple spatial scales. So we can kind of look at how conditions in San Mateo County compare to conditions in Monterey. So what is a watershed? It's kind of a word that we throw around a lot. A watershed is really, it's just a spatial area of land that all drains into the same body of water. So if you imagine a droplet falling, at the crest of the mountain, and it takes the journey all the way down to the ocean, across land, into streams, into creeks, into rivers, maybe even, and eventually the ocean. That water can carry pollutants on land into the ocean, which means that actions on land are directly connected to the water quality downstream. For us in San Mateo County, we have several coastal watersheds that do drain into the Pacific Ocean. And San Mateo County actually has a very interesting sort of interactive watershed map. And we'll drop that in the chat at the end. So you're not all tempted to look at it now, but it's very interesting. So we'll drop that in there for you. So this program is made possible through funding and donations from our partners and sponsors. Our partners for this program include the San Mateo County and, as I mentioned, the Monterey Bay National Marine Sanctuary. And funding comes from our sponsors, which include the City of Pacifica, the Rose Foundation, Sewer Authority Mid-Coast Side, and San Mateo County Harbor District. So we thank all of them for making this happen. But most importantly, this program is made possible by all of you. So we're extremely thankful for the volunteer community scientists that make this project happen year after year. It's a huge commitment, but it also pays dividends for us just to be able, and everybody, to be able to have this data, to be able to continue this program for as long as it's been going. So, big kudos to all of you. Thank you for joining us. So we talk a lot about water quality, similar to watersheds. It's kind of, a term that we throw around. But what does it really mean? Um, the water quality parameters, and we'll jump into these now that we're studying in the First Flush program, include fecal indicator bacteria. Sometimes we call it FIB. Nutrients, which for this program we sample nitrates and orthophosphates. Metals. For this program we sample copper, zinc, and lead, and total suspended solids. And we'll get more into that, but it's essentially a measure of how much sediment there is in the water. And then lastly, in our data sheets, we'll have places for you to make other kinds of observations like smell or, if there's wildlife around. So site conditions and observations feed into this program as well. All right, so the first parameter that we'll talk about is fecal indicator bacteria. Indicator bacteria serve as an indicator for the presence of the feces of warm-blooded animals, which indirectly indicates the potential presence of pathogens such as viruses and bacteria that can impact human health. So there are primary and secondary sources of FIB. Primary sources include humans, waste from humans, domesticated pets, and wildlife. So it includes everything like deer and birds and all of that. And then secondary sources include areas where bacteria may fester or proliferate in the environment. And so that can include like sand or sediment that sort of water is running through and then being stagnant, or in biofilms, which is sort of, an algal bacterial growth that can occur in standing water. If a stream or waterway has high levels of FIB, it's possible but not guaranteed that this water could make you sick. You may notice I'm using a lot of qualifying language like may or possible or, things like that. And that's because the science behind evaluating public health risk from fecal indicator bacteria is extremely complex. But what's important to remember is that the thing we're measuring here, which is E. coli and Enterococcus, are not necessarily the bacteria that do make you sick. But their presence does indicate the possibility that there are things in the water that can. So it's a little bit confusing, but that we use these samples as sort of indicators that there may or may not be poop in the water. Our next group of parameters that we look at for the First Flush program are nutrients. So nutrients are essentially plant food that are essential to plant growth. Fertilizers that contain nutrients are therefore essential in the context of agricultural production, residential gardens, and ornamental landscapes. Agricultural fertilizer use is regulated at the state level under the Irrigated Lands Regulatory Program. A little bit of alphabet soup there for you. Whereas residential use is not regulated other than labeling requirements for these products. Other sources of nutrients can include some pesticides, detergents, and human and animal waste. Elevated nutrient levels in streams and oceans can cause algal blooms, and you may have heard of this. But when you have a really large algal bloom, that can sometimes lead to oxygen depletion in the water when the excess algae decompose and break down in the water. They eat oxygen, and then you're left with water that doesn't have as much oxygen in it. This process, which is known as eutrophication, kind of a $1 million word there, can lead to large-scale fish die-offs or other impacts when dissolved oxygen gets to dangerous levels. Essentially, the algae eats all the air and the fish can suffocate in large numbers. And you'll see that in the picture on the right here. Our next set of parameters are heavy metals. And so heavy metals can enter waterways from gutters or roofs, brake pads, tires, industrial waste, treated lumber, paint, fires, and a whole list of other natural and human-related sources. Heavy metals can be essential micronutrients, though, so it's a little confusing at appropriate doses. Um, but at high doses, they can be linked to ecological health impacts or impacts to human health. For example, some of us may take zinc supplements to help boost our immune system, which is perfectly healthy and safe. You do want to follow the bottle's recommendation for how much of that to take. But on the flip side of that, as we all know, ingesting high amounts of lead, for example, can cause serious health complications, especially for young children. So the three heavy metals that we test in the first flush program are copper, lead, and zinc. And so the last set of parameters that we'll talk about today, I mentioned earlier, is the total suspended solids. Total suspended solids, also known as TSS, are particulate matter or sediment in the water that attract charged particles, which can include pesticides and metals. TSS can originate from natural or accelerated rates of erosion, including things like construction sites, agricultural runoff, fires can definitely cause high levels of TSS, and other sources. TSS can impact the environment through attachment and mobilization of pollutants. So, as the water's rushing through, those sediment particles in the water can have a charge to it that actually attracts those pollutants. And when we say mobilizes them, that means it picks them up and brings them with it in the water. TSS are often associated with the presence of other contaminants such as bacteria or heavy metals. And if anybody has questions about any of that, happy to discuss it at the end. But this is sort of the foundation of, why we do it, what we're doing, why we do it, and a little bit about, what water quality parameters we're focused on. We can answer questions at the end, but also we want to be a resource to you to learn as much as you want or can. So, please don't hesitate to reach out to us. We'll provide you with our email addresses and everything. So, we're happy to talk about this throughout the whole thing. We want it to be a learning experience for everyone. And with that, I will kick it over to Grace. We're getting into the section more focused on logistics and things like that. So take it away.
Awesome. Thank you, Clifton. And yeah, thank you all for being here. We really appreciate your support in this program, that you are the heart of this program. And so what does it mean to be a First Flush volunteer? So that means that you're going to be an important part of community science and engagement. And you're going to protect water quality for the health of people and wildlife. You're going to go out in the rain and the elements to collect water samples and collect field notes. And this might be in the dark, it might be in the middle of the night, it might be, well, it's certainly going to be raining. So we'll be prepared for that. And then you're going to be on call 24/7, sort of, and we're going to go into that, but we don't really know when the first flush event is going to be. So it could be anytime in the wet season. So we have to kind of keep an eye on the weather, and we're going to be as communicative as possible with you guys. And it's really a huge part of this, and where you all come in, is that it's really hard to capture this many sites along the coast around the same time, kind of capturing similar circumstances or elements. So we do this by having you guys help us, and also splitting up into 5 teams. So you can see that team 1 is furthest north near Pacifica, and team 5 is the furthest south near Half Moon Bay. And so we're also going to have team captains, and the team captains are going to coordinate with their team and with us to kind of decide where you meet, who's going to pick up the materials and all of that good stuff. So we're going to have you fill out a Google form, and that's the QR code for the link. And then we're also going to drop the link in the chat. So you're going to, in that form, select which team is your preference, your first and second preference, as well as kind of talk about your availability. So like what times of the week are you most available? If you're going on any major vacations or holidays and you're not going to be in town, then we won't contact you during those times. Um, but yeah, so just select your preferences, say whether you'd be willing to be a team captain or not, and also note that we might not be able to give you your first or second pick depending on availability, but we'll definitely like work with you to make it as convenient as possible for you. So yeah, we'll give you like about 5 minutes to fill that out. But yeah, and then we'll drop that link in the chat so that's easier for everyone. All right.
System failure. I'll bring that up in just a second. I'll stop sharing until I.
Okay, I did drop the link in the chat, so we should be good.
Oh, nice. Thank you.
Yeah, I should. Yeah, and we'll also send out an email after this presentation that will include some of the links that we mentioned and that form in case you aren't able to fill it out during this time. Um, yeah, so and so team one is going to be closer to Pacifica. So for this north, and then team two is going to be um, kind of a little further south, and then team three is near kind of Pillar Point Harbor area. And team four is going to be a little further down, and then team five is in Half Moon Bay. All right, I think we'll continue with the presentation. And again, if that time wasn't sufficient, we'll be sending it later, or you can fill it out during the presentation.
All right.
Awesome. So how does this work? So we're going to keep monitoring the weather and give you a notification when we think the event might happen. And we're going to try to give you as much notice as possible so that it's not spring upon you. But with that being said, we can't control the weather. I wish we could. So we'll try as hard as we can.
We'd all be rich if we could.
Yeah. Awesome. And so the team captains, which you're choosing with you whether you would like to be one or not, are going to coordinate picking up the materials from the RCD office, meeting for sampling, and then also dropping those samples and sampling equipment back at the RCD office. So yeah, you're going to coordinate with your team and with the RCD. And again, we're gonna try to monitor the weather as much as possible, but it is possible that we'll have a false start where the forecast looks like it's going to be the event, but then the rain conditions don't actually match what was forecasted. And this did happen last year. So brace yourselves, and also we'll try our best to prevent that from happening. So on the day of the first flush event, you are going to wait for the alert. We're going to try to give you as much heads up as possible. And then whoever is going to grab the materials from the RCD office is going to meet us there, and we'll provide those to you. Then you're going to travel to your team's assigned sites, collect your samples, and fill in your field sheets. And then after all of that, you're gonna have someone bring the materials and the samples back to the RCD office. And we're going to provide you with all of the supplies that you might need. And this is a list, but I'm going to go ahead and show you all in person. Well, not in person, over screen.
Let me just pause the presentation here. At least stop sharing. How do we do that? Oh, here we go.
Awesome. Okay, cool. So we are going to provide you with a cooler that's going to have ice in it. That's a little too big for me to hold up, so I'm sorry about that. And then we're going to provide you with a sampling bucket. So that's right here. The smaller one is the one that you're going to collect your samples in. And at some of the sites, you're going to lower it down with a rope, but not all of the sites require that. And then we are going to provide you with some smaller red solo cups they're going to use if the flow isn't strong enough to collect with the bucket. So those are going to look like your standard red solo cup. And you're going to use these to collect the water and then put it in your sample bucket. And so for both of those supplies, you're going to clean them in between each of your sites to prevent contamination of your sample. And then also we're going to provide you with paper towels because you're going to be in the elements, it's going to be raining, it's going to be messy, and we want the best experience for you as possible. So we're going to have those for you. And again, we don't know when the event is going to happen. It could be late at night, but it is going to be raining and we want to protect your safety. So we will provide you with yellow rain jackets, but if you have your own rain jacket, we have high-vis vests as well. The water containers. Yeah. So we're gonna have distilled DI water, and that's going to be sanitized, and then we're going to have alcohol, and so we're going to go into the cleaning procedure a little bit further in the presentation, but yeah, we're definitely trying to keep everything as sterile as possible for our samples. So yeah, we're going to provide you with high visibility vests and cones so that you can put those out while you're sampling. And then we're going to provide you with whatever size of glove you prefer. So you're going to wear these gloves to prevent contamination of the sample and also protect your own health because we don't really know what's in the water. So I want to keep you safe. And then we're going to provide you with a clipboard and your field sheets. And so these are going to be Write in the Rain paper. And you're going to want to use pencils on this. And Write in the Rain paper is a type of paper that doesn't get wet, essentially. It repels the water. And so you're going to take your field notes on these, and we'll talk about those a little bit later. And then we're going to give you a bag with hand sanitizer and headlamps and some Sharpies, some pencils, and a pencil sharpener. And you're going to use these. And then all of these supplies are going to come in a larger bucket. So you're going to have a smaller bucket for sampling and then a larger bucket that's going to hold all of these. And I think that's all of the supplies. Sweet. Yeah, so these are the steps for the sampling. So step one, you're going to put on your gloves. And then step two, you're going to label your bottles. Step three, you're going to fill out your field sheets. Step four, you're going to clean your bucket or your cup. Well, you're going to clean both. And then step five, you're going to fill all of your sample bottles. And then step six, you're going to place the samples in the cooler with the ice. Yeah, so this is step one because you want to put on your gloves to again protect your health and your safety and also prevent contamination of the samples. And then you're going to label your bottles with the date and time. There are going to be 4 bottles in these plastic bags, and they're going to be labeled with your field sites and with the sample IDs. And then the only thing you're going to need to fill out is the date and time. And the time doesn't have to be exact. Like if it's 6:02 a.m., you can say 6 a.m. But what's really important is that it matches your field sheets and is consistent. Because if it's different, that can cause some confusion within the samples. And then another thing to note is that these are going to be way easier to write on before they get wet. So I would recommend doing that in the car before you go out to sample.
Yeah, trying to write with Sharpie in the rain is never fun.
Yeah.
You might get a couple of numbers in, but then just nothing. So yeah, it's definitely good to do that in a dry environment.
Mhm. Awesome. So this is an example of the field sheets. And yeah, these are going to be right in the rain paper. So it's easiest to use pencil because sometimes the pens or Sharpies might not work on these. And so you're going to fill out date, time, your site, your team members. You're going to fill out also, oh, very important, make sure to note all of your field observations because you guys are our eyes and ears out on the field. And so it's really, really helpful if you note anything that you notice. Because when we're analyzing the data, it's going to help if we like look at the results and we know kind of the circumstances of the site at that time. So nothing is too small to write down. So truly anything you notice that you want to write down, feel free.
Yeah, and we'll be out there with you, but there's only 2 of us. So there will be a few teams. So yeah, we really appreciate all the observations that you guys can provide with us.
Awesome. So we're going to go over the procedure to clean the buckets and the fill bottles. So again, it's very, very important to clean your bucket in between sites because you want to have prevent contamination of the sample and make sure that it's a representative sample for the creek or the outfall. So first, you're going to rinse your sampling bucket with the squirt bottle of alcohol. And you're going to go kind of like around the rim, trying to cover as much surface area as possible. And you're going to then dump that alcohol and you're going to repeat this 3 times. And then after you've done that, you're going to do the same thing with the water squirt bottle. And so you're going to go around the rim of the bucket with that and then kind of like twirl it around and then dump that. So again, you're going to do that 3 times. And then finally, you're going to rinse your sampling bucket with creek water 3 times. So you're going to like collect your water and then dump it downstream and do that 3 times. And it's really important that when you're dumping these, that you're going to do it downstream of your sample site, because we want to prevent contamination of the sample, and we don't want to stir up more sediment into the water column. And if the flow is too low, you're going to use your red cup to rinse the little bucket 3 times with sample water. And then you're going to use the red cup to fill the sample bucket. Cool. Yeah, and then you're gonna fill your sample bottles with sample water from the sample bucket.
Yeah, and it looks like we have a question from Sydney. Where do you put the used alcohol?
You can dump that. It's very low quantities of alcohol, so you can just kind of dump it downstream.
Yeah, and it does evaporate out. I mean, even in a storm.
Mhm. Awesome. So we want to prevent any contamination of our samples. So we want to make sure that we're following the procedure as well as we can. So we're not going to touch the inside of the cups, the buckets, or the sample bottles. And this is just a measure to make sure that our samples are representative of the streams and the conditions.
And actually, I'm gonna hop back. I feel like this picture on the bottom left is a good example because sometimes there will be enough flow that you can just dump the bucket, the blue sampling bucket in there. But this is a good example of when there's not really enough flow to do that. And so, if you tried to do that with the bucket, you'd mostly just be scooping up sand. So that's really where the red cup comes in.
Yeah, that's a really good point. Thank you. Awesome. So one team is going to take a duplicate, and this is just a way to measure the the representativeness of the samples. So you're going to fill your original sample bottle for that site first, and then you're going to fill the duplicate sample bottle using the same bucket of water um, for the duplicate. And then we're also going to take a field blank, and field blanks are sample bottles filled with distilled water. So you're going to fill the sample bottle marked blank with the distilled water bottle, that's like the squirt one, provided in your larger bucket. And then same with the rest of the bottles, you're going to label with the date and the time.
Awesome. Yeah, and both of these are sort of quality control measures so that we can know, it tells us if we can trust the data or not.
Mm. Awesome. So when you're sampling a creek, you want to sample where water is flowing. We don't want to collect where there's stagnant water. And you want to try to get as little sediment in the sample water as possible. So like if it's turbid, we understand that that's a marker of the water, but try to avoid like picking up sand or dirt when you're collecting your sample. And then when you're rinsing your bucket, make sure to throw your water downstream. Again, as to not stir up sediment and also to not contaminate the sample. And when you're sampling a stormwater outfall, you want to sample where water is flowing directly out of the outfall. And you don't want to sample if the high tide is mixing in. And both of these are so that we can actually get the water that's coming from the land, from the outfall, rather than the marine water mixing in. And it's very important to not stand in front of the outfall, just as a safety measure. Um, and then I think there are some pictures on the next slide of like where you can stand.
Oh, cool.
So, yeah, so in these situations, you're going to use your bucket with the rope, and these folks are sampling from safe places. All right, so to recap, at each site, you're going to firstly put on your gloves, then you're going to label your bottles with the date and the time inside a dry spot, and then you're going to fill out your field sheets with as many observations as you can. Then you're going to clean your bucket and cup, fill up all of your sample bottles, and then put those samples in the cooler with ice. Awesome. And your safety is our priority. So these are just some tips for when you're going out sampling. So you always want to be aware of your surroundings, just kind of be cognizant of like what's going on, where you are. And we never want you to risk your safety. If anytime you feel unsafe or like the conditions are a little spooky, just go to your next site, say that it was not good conditions to sample. Um, your safety is worth way more than a sample. Um, don't put yourself in front of rushing water and don't walk on unstable ground. On in storm conditions, it's could be very easy to lose your footing. And if you're in water, that's also kind of scary. Be aware of wildlife and protect yourself from exposure to the elements. So it's going to be raining, it might be cold, you're going to be around water. So we want you to wear your gloves, wear warm attire if it's cold, wear good footwear, wear your high visibility vests, and never sample alone. It's really, really good to have your team there just in case something happens, in case you might like lose service. It's really important to have a buddy. So what are the next steps? We're going to have our in-person sample training Saturday, September 21st at 10:30 a.m. And we're going to meet at the Fitzgerald Marine Reserve parking lot. But if that time doesn't work for you, we're also going to have video training for that. We have a little walkthrough with how to collect a sample, how to clean your materials, and all of that good stuff that we'll be able to send you. And then after you fill out that Google form, we are going to send you an email of what team you're on, and we're going to send you the site directions. And then once you get those, please review your site directions and visit because it's way easier to go visit in the daylight when it's not raining. So then when it comes to the first flush event, you'll kind of know where you're going and have your footing. And then we'll also be sending you the sampling instructions that you can review so that it's fresh in your mind when the first flush event happens. And then we're going to wait for the rain. So you guys are going to be on call until it happens. We're not sure when it's going to happen, but we will be in contact with you when it does. It has happened on Thanksgiving. It could be in the middle of the night. So we're really relying on you all to help us with this.
But last year was like a regular time, right? It was like 6 p.m. or something.
Yeah, exactly.
So
Yeah, it was.
it's not always at the worst possible time, but you
Yeah,
know.
exactly. Yeah, it was a Friday at 6 p.m. So it was honestly quite nice.
Oh, there we go.
Cool. Awesome. And then when the first flush event does happen, we're going to give you a call, or rather, your captain will coordinate with you. You'll coordinate to meet and pick up the supplies for sampling. You're going to go out to your sites and collect your samples. And then someone's going to return those samples and the supplies to the RCD. And the RCD office's address is down there. All right, and then to recap, you're going to want to visit your sites before the first flush event occurs. Again, just to make sure that you're familiar with where they are. And to be honest, they're all very beautiful, so it's a nice trip to go on. And then you're going to coordinate to meet at the RCD office once the first flush event is going to happen. And then you're going to drive to the sample sites, fill up your sample bottles, fill out your field sheets, and then return those materials and the samples to the RCD office, and then we'll take it to the lab. And importantly, have fun. This is a really fun program. You get to be a scientist, you get to collect samples in the dark, and it's a really great way to connect with your community and just feel connected to science too. It's a really fun time. I had a blast last year. And again, we're not really sure when First Flush will be. These are some of the historic dates. As you can see, last year it was November 17th at 6 p.m., which was very convenient, and I was appreciating that. But it can range anywhere from September to November. So we're not really sure, but yeah, we'll try to be as communicative as possible when we see it in the weather forecast.
Yeah, and I love this slide. Like it's really interesting to kind of look at the different times of year that it happened. Um, And one of the things with this program is that we have these sort of rain triggers. So when we're watching for storms, there's certain things that will trigger the event. One of them is a certain amount of rain. I think it's 0.1 inch, which is not very much. So another trigger is that we're really looking to have rain sort of sheeting off the roadways and really picking up all of the stuff that may or may not be there. Um, so for example, I was looking at the rain. We're already keeping our eyes out, and we are going to have a little bit of rain like Sunday night, Monday morning. And there's a chance, it'll be a little bit close to that 0.1. But since it's so early in the season, for example, we're not super likely to have those sheeting conditions. So, like we said, we can't predict it 100%, but we're always going to be keeping an eye out. And I have a feeling this weekend is probably not going to be the one, just because it's so early in the season. Things just aren't really soaked in yet.
Awesome. So with that being said, thank you so much for being here. You are the heart of this program. We couldn't do it without you all. And if you have any questions, we're going to up the floor to questions in a little bit. But you also have our contact information, so you can email us at firstflush@sanmataeorcd.org or go to the RCD website and you can find our contact information there as well as our social media.